Good afternoon, everyone. It's on, right? Good afternoon, everyone. Today I have the pleasure of introducing Tracy Centanni. She comes to us from Boston to talk today about dyslexia and some of the work that she's done. And to introduce that work, I'd like to introduce her a little bit. She did her undergraduate at Penn State University and spent a lot of time in Michael Wenger's lab doing various types of cognitive neuroscience. Very interested in the neuroscience of memory and cognition and spatial patterns and stuff like that. So I gave her a good firm foundation for doing graduate work in a neuroscience program with Michael Kjellgaard in Dallas, at UT Dallas. In this lab, and what she did her dissertation on was working with animal models for dyslexia. And you might think, well, animal models for dyslexia, I won't take away any of her punchlines, but it is possible to do some work that is quite relevant to dyslexia in an animal model. Obviously, they don't read and comprehend the way we do, but there's component processes there that can be unpacked quite nicely in a model system. And so she did her dissertation on that. She then changed gears a little bit and did a postdoc with Tiffany Hogan at Massachusetts General Hospital. And Tiffany Hogan is a speech-language pathologist who also does a lot of work in developmental psychology, specifically in language and reading. And so Tracy has moved into a little bit more clinically relevant, sort of directly clinically relevant, working with kids and moving more into that direction. Currently, she's at MIT, and she's doing neuroimaging on children, and she's interested in branching that back into the animal models so that we can get a nice loop of animal work on specific genes, taking that to hypotheses about the brain, testing those hypotheses about the brain in kids in clinical populations, and then going back and refining again with the animal models, sort of all in a coherent loop. So her training is enviable and quite varied, and she comes to us with big ideas, and more importantly, she comes to us with data. So that's what she's here to talk about. So thank you so much.
Do I have a mic? Everyone can hear me okay? In the back? Yes? Okay, great. So thank you, Chris, for that very nice introduction, and thank you all for coming to hear me talk today. I will talk loudly over the crunching of chips and chewing of sandwiches. So again, today we're going to talk about auditory processing in dyslexia and the way in which we can use animal models to inform our experiments in humans with clinically relevant speech and language disorders. And so we know that language is one of the skills that makes us uniquely human. It's something that we need to interact with everybody else in our world. Reading, speaking, sign language, all of these things are instrumental in our daily lives. And children that are born with communication disorders are born at a distinct disadvantage. And a lot of these communication disorders, for example, dyslexia, are not diagnosed early enough in life for sufficient training. So unlike many medical conditions, disorders like dyslexia, again, are only diagnosed after failure. So in the case of dyslexia, it's only diagnosed really in grades 2 or 3. So after a child has already had several years of reading instruction, if the child is still struggling to read, that's when they're diagnosed. So we're really missing so many years in which we could be intervening in these kids' lives to help them be better readers later on. Of course, the core neural problems that are present in these disorders exist from birth. So we know that communication disorders are prevalent. Approximately 7 to 15% of kids have dyslexia, and they're also highly heritable. So for example, in dyslexia, there's about a 68% concordance rate of dyslexia in monozygotic twins. Of course, we don't know what the causal gene is for dyslexia, and chances are there isn't one. There are several disorders that we know of that are single gene disorders. For example, Fragile X is a single gene disorder. If you inherit the mutation in the FMR1 gene, you become a carrier for Fragile X. It's a one-to-one relationship, and it's easy to test genetically for this disorder early in life. So for example, if we look at a family tree of inheritance for Fragile X, you can see, again, the males are in squares, females are circles, affected individuals are black symbols, and unaffected individuals are white circles. So in this case, we can see the mother in this family is a carrier for the FMR1 mutation gene, and her children that inherit that mutation also inherit becoming a carrier for Fragile X. The problem is that dyslexia is not so straightforward. There is no single causal gene for dyslexia, and the genes that are involved in dyslexia vary greatly across different individuals. So there is not an easy genetic test for dyslexia and probably will never be. So what is dyslexia? I won't read this entire definition to you, but the basic take-homes are it's a neurobiological disorder. It's characterized by difficulties with word recognition, spelling, and decoding. And these difficulties are thought to be driven by an underlying deficit in the phonological component of language, so the smallest building blocks of a word. And this disorder, again, is present in kids with normal or above average IQ and with adequate schooling. So this is not a problem of a learning disability. This is not a problem of inadequate education. This is a neurobiological problem in phonological processing. And this disorder is currently being lumped into this bigger umbrella of learning disabilities across the nation. So it's thought of as a subtle disorder that doesn't really deserve its own label. But in fact, these kids have large consequences throughout their life, including lower academic and vocational success, low self-esteem, and delinquency in adolescence and adulthood. And this is a little bit of an outdated study, but in the year 2000, there was a study published showing that almost half of the incarcerated inmates in the state of Texas have dyslexia. So it is definitely a big problem affecting our youth. So what can we do? We know that earlier intervention helps. Of course, by definition, since dyslexia is not diagnosed until grades 2 or 3, there is no formal intervention for younger kids. But there's plenty of anecdotal evidence in parents with either older children with dyslexia or a rich family history of dyslexia that provide some extra support for their younger children as they grow up, and those kids tend to grow up in the normal reading range. So there's anecdotal evidence to suggest that support prior to the onset of reading instruction actually may be helpful. And if we had a reliable genetic or neural profile for this disorder, we would be able to not only diagnose earlier, but hopefully increase intervention success. So our current approach to digging into the genetic and the neural issues in dyslexia typically use genome-wide association studies. And so these studies take hundreds to thousands of individuals that share a phenotype and look for common variants in the genes that show up more often than you'd expect by chance in these populations. And they're great because we've dug into a lot of different genes that show up often in individuals with dyslexia, but of course these are correlational studies. So we don't really know which genes popping up in genome studies are just associated with the disorder or whether they're actually causing the phenotype. There's also no way to isolate individual genes. So if we have 8 to 10 candidate dyslexia genes, what is each of those genes doing to contribute to the overall phenotype that we see in dyslexia? So we need a model in which we can control the genetics and also test for speech-sound processing differences. And Chris already gave away my first punchline, but that is that we're going to use animal models to do so. So we can use rats, and we have precise genetic control in these animals. And not only can we control the genetics, but these animals are actually a really good model for speech-sound processing. So we can train a rat to discriminate human speech sounds. They can do consonant discrimination, so they can press a lever to the sound dad and ignore bad, gad, sad, tad, et cetera, et cetera. They can do vowel discrimination. They can do the task in quiet, in background noise, in various levels of degradation. They can do it at a variety of speeds. They can generalize across speaker and gender and so on and so on. And their performance curves on these tasks are very similar to what we see in humans. So they can handle the same amount of background noise we can. They can handle the same speed that we can. So at the very basic auditory processing level, this is a great model to study speech-sound processing on the neural and the behavioral level. And so I'm going to show you briefly what one of these tasks might look like. Hopefully we have adequate sound here. Oh, of course it's not finding my media. Well, okay, for the sake of time, we'll skip the video, but I have it after if anybody wants to see it. I apologize for that. Okay, so animals are, again, a good model for speech-sound processing. However, again, rats don't read or speak English. So, of course, we're not training them to read. We're not training them to speak our language. But at the very basic auditory processing level, they process speech sounds very similarly to the way we do. Another problem is that genes may have different functions across species. So even though we have rat homologs of all the genes we're studying in humans, the way this gene reacts in the brain of a rat might be different than the way it reacts in a human. So neither method is really sufficient on its own, and we really need to get into this pattern of using these two models together to inform our study of dyslexia. Okay, so we can use both of these models to make real progress on the hunt for disorder mechanisms. And to that end, I'm going to present today 2 experiments that are moving us in this direction. First experiment I want to share with you today is work from my dissertation in which we used a rat model to determine the role of a specific dyslexia gene on speech sound processing. So there are two questions that we need to ask. First, why are we looking at sound processing? This is a reading disorder. And second, how do we make a rat model of dyslexia? So first, let's talk about auditory processing. So if you think back however long ago it was when you were learning how to read, your teacher looked at a sheet of paper and said, this is the letter D. It makes a duh sound. This is the letter B. It makes a buh sound. So right from the beginning, you're taking the auditory representation of a sound and mapping it to the letter on a page. We know that individuals with dyslexia have abnormal neural responses to auditory stimuli. First is the diminished mismatch negativity. So this is a feature in the brain where if you play the same sound, for example, ga, over and over again, the amplitude of the response to each successive sound will decrease. So the brain is basically habituating to the same sound being presented over and over again. If you then present a new sound, for example, da, the amplitude of the response returns to the initial response, basically marking that there's a change that's happened in the stimulus. In kids with dyslexia, this response is much smaller. The difference in these peaks is significantly smaller in kids with dyslexia, suggesting that the brain of children with dyslexia is less able to mark these changes in the auditory signal. Another well-replicated auditory processing difference is in the response latency and the amplitude of the response. So for example, over here we have EEG traces in response to tones in individuals with language impairment and controls. And you can see, for example, T4 shows this effect very nicely, that in the dark lines of our language-impaired kids, the latency of the response is not only delayed, but the amplitude is significantly lower in the affected individuals compared to controls. And so at the very basic auditory processing level, there are very consistent steps across species. So by the time sound goes through the ear, passes through several subcortical areas before it reaches primary auditory cortex. Now, this is the first place in cortex that sound reaches before it goes on to, in humans, our other language-related areas. But up to primary auditory cortex, all of these steps are the same, very well conserved across species. And so things can go wrong at any of these stages. And if there is an abnormality in the primary auditory cortex area, we'll be able to see this in the rat model just as easily as we would be able to see it in a human. All right, so how do we make a rat model of dyslexia? In the mid-'80s, there was some work done by Galiberta and his colleagues looking at the brains of individuals with dyslexia, trying to figure out what is different in these brains compared to typically developing brains. And they found a lot of cortical abnormalities that they could physically see in these brains, one of which are ectopia. So for example, here's a slice of cortex. The neurons are stained dark. And you can see that in normal cortex, neurons are pretty evenly distributed. However, in these individuals with dyslexia, they found ectopia, or clusters of neurons in a layer where they don't belong. This is a very obvious physical sign of this individual being affected with dyslexia. And when we started getting the genetic techniques to dig into what's causing these ectopia, we found 4 neural migration genes that were significantly linked to dyslexia. So if you think about cortical neurons clustering where they don't belong, this is a feature of abnormal neural migration. So these are the first 4 genes that popped up. And we can replicate these cortical abnormalities in animal models. So if you suppress any of these genes in a rat or a mouse model, you end up with clusters of neurons exactly the same as what you see in humans. So on the physical level, it's very comparable. We know that the gene KIA0319 has a role in dyslexia. There is one study to date, published in 2012, looking at the relationship between a SNP in this gene, or a small little variant in this gene, and auditory processing of language and reading. So for example, in individuals with a SNP in the KIA0319 gene, it's linked to reduced activation in the left hemisphere language areas during a reading task. So already we've found that there is a link between this gene and specific reading and a specific neural marker. The question is, does it interfere with auditory processing? And so for the first study, we wanted to know if suppression of this gene, the rat homologue of KIA0319, would interfere with neural processing of speech sounds. Okay, so for our first study, we had 11 animals that we anesthetized. We acquired multi-unit recordings from primary auditory cortex. 5 of these animals had suppression of KIA 0319 using RNA interference. Basically what this is a marker, so basically a tag, so that when the RNA is being transcribed into protein, basically this gene gets skipped and this protein doesn't get made in those cells. And this is not a complete knockout, so some protein is still being made in the brain, which is an important distinction, because that is typically what you see in humans with dyslexia. So individuals with dyslexia with a variant in this gene tend to have reduced protein expression. It's not that they have no protein, but that the protein levels are reduced. So these animals heard a variety of auditory stimuli. We played some broadband click trains at a variety of speeds. We played some isolated speech sounds, and then we played a tuning curve to determine the characteristic frequency. So in the primary auditory cortex, every neuron has a frequency that it likes to respond to the best, or its characteristic frequency. And so we can map the auditory fields based on each characteristic frequency. So for example, if we play a variety of different frequencies at a variety of different intensities, and we mark the action potentials of each site in these black tick marks, you see kind of this inverted triangle function. So when the sounds are played very loudly, this site responds to pretty much every frequency. But as the sounds get softer, it gets more selective in which frequencies it responds to. And so the lowest point of that triangle we refer to as the best frequency or the characteristic frequency, where the neuron responds to the best. And so, for example, this site's characteristic frequency would be around 6 kilohertz. And the characteristic frequency of neurons is organized in the brain. So, for example, here's the primary auditory cortex of a rat. And you can see the color of each little tessellation marks the characteristic frequency of that site. So, for example, the sites that like low frequencies tend to be grouped together, followed by the mid-range frequencies, followed by the high frequencies. And this tonotopic organization is well preserved again across species. So at the level of A1, responses to speech sounds are the same in rats as they are in humans. So we have our knockdown animals, and the first thing we want to do is validate our model using a technique that mimics what we see in humans. So for example, here's the same EEG trace I showed you before in humans in response to tones. Our dark line is our affected group. And in the animal model, we do have electrodes in the brain, but we can also gather local field potentials. So activity that's summed over a wide number of neurons, which closely mimics the technique and the type of data acquired by EEG. And so what we found in the LFP response is exactly the same pattern we see in humans. So in the dashed line, we have our knockdown animals. The solid line is the controls. And you can see, again, this delayed latency and this reduced amplitude in the response. So on the local field potential level, our model mimics what we see in humans with dyslexia. Now, when we read a lot of these papers in the human world, they refer to this lower amplitude as reduced expression, hypoactivation, or reduced activity. But because they don't have electrodes in the brain, you can't actually validate this hypothesis, whereas we can here. We have action potentials in the brain, we have electrodes in the brain, and we can count action potentials. So we did just that. And we expected to find that these KIA knockdown animals would fire fewer action potentials in response to sound compared to the controls. And actually, we found no difference. And we were very surprised at this. So within the consonant part of the stimulus, as well as in the vowel part of the stimulus, there's no difference in the amount of times neurons fire in the knockdown animal compared to the control. So if the number of action potentials is the same, what's causing this reduced amplitude? So we looked at the timing of those responses. And what we found is a significant difference in the timing of those responses. So if you calculate onset latency, the time between the stimulus onset and the time at which the neuron fires, there is significantly higher variance in the timing of the onset response in the knockdown animal compared to the control. So that when you average across neurons to get this LFP, you have the illusion of a lower amplitude. Does that make sense? Okay. We can see this also if we look at single site responses. So this is a control site responding to 20 repeats of the sound B and 20 repeats of the sound D. So each response is a row. They're stacked on top of each other. So here you have 40 repeats of these sounds, and each of these black dots is an action potential firing in the brain. So in the control animal, we can see that in response to D across repeats, this neuron fires very quickly and very consistently. Whereas in response to B, it waits about 10 milliseconds and then fires. And if you put these data into a nearest neighbor classifier in a computer, it gets it right almost every time. The only time it gets it wrong is where I have this X here, where this neuron fired a little bit too quickly, and the classifier misclassified that as a D response. So you can see these responses are very consistent. If we look at a comparable response in the knockdown animal, you can see not only this delayed latency here, but the increase in the variability of when that site's going to respond to both D and B. And if you put these data into a classifier, it's almost at chance at 67% at being able to tell which sound we played that drove that individual response. And this was consistent across all the consonant pairs that we tested. One more way of visualizing this, these are now average responses across neurons. We call these neurograms. And you can see the characteristic frequency of each site on the y-axis, and then time in milliseconds on the x-axis. So in our top panel here, we have all of our control animals. And you can see that there are very unique patterns that are evoked in auditory cortex in response to consonant sounds. Whereas in the Kia knockdown, these responses are much more spread out in time. So you can imagine that it might be more difficult for the knockdown animal to tell these sounds apart because the patterns in the brain are so similar. So to test the effect of this neural feature on behavior, we grabbed a new group of animals, 26 rats, 16 with the knockdown, and we tested them on a variety of speech-sound discrimination tasks. So they did a full speech task where they pressed the lever to dad and ignored bad, gad, sad, tad, dud, deed, and dude. They did a speech and noise task where they did the same discrimination task in 4 different levels of background noise. They did a speech sequences task where they had to locate the sound dad in a continuous stream of distractor sounds at a variety of speeds. And then we did a truncated speech task where they only received the consonant information. So we'll talk about that a little bit more in a bit. And then we mapped the brain at the end of all training. So first we trained them on this full speech sound discrimination task. So again, pressing the lever to dad and ignoring a variety of distractors. And you can see that in the first 5 days, there's already a significant difference in behavioral performance where the controls pull away quite quickly compared to the knockdown animal. And it's not that they're responding to the target less, it's that they're false alarming to the distractors significantly more. And we did test reaction time because we wanted to make sure the animals weren't just sitting there jamming on the lever and being confused, and there was no difference in response times. So it's not that they're sitting there confused, just jamming on the lever. We think that they're genuinely not sure which sound is the target sound. So I mentioned a little earlier that our knockdown technique has the benefit of not being a complete knockout. So some neurons are affected by the transfection, some neurons are not. And the number of neurons that are affected varies between individual animals. So after the animals were through the experiment, we could go back in and actually quantify how many neurons were affected by the transfection. And the amount of transfection in the knockdown animal directly correlates with their performance on this task. So it does seem like the amount of Kia 0319 protein in the brain is directly related to behavioral performance, and we saw no relationship in our controls. So if we look again at these first 5 days, we can see that even though the knockdown animals are impaired, they do seem to be getting better over the course of those 5 days. So it could just be that this is a delay in learning, that maybe if we gave them more time, they would eventually catch up. So we gave them 5 extra days, and lo and behold, they do catch up. By the end of 10 days of training, the Kia knockdowns are performing just as well as the control animals. So perhaps this is just a learning problem. Perhaps if we gave them a more difficult task, they'd be just fine. So we gave them a speech and noise task, which is a task that's known to be difficult for children with dyslexia, and they fell apart again. So even though they learned the task just fine and quiet, albeit after a delay, as soon as we introduced noise, they were significantly impaired compared to the controls. So it could be now that task complexity is the problem. So maybe if we gave them a rapid speech task, they'd be just as bad at rapid speech. But they're not. So throughout the entire rapid speech task, there were no differences across group. So it seems to be that the speech and noise task is a very specific deficit in these animals, and that the full consonant vowel consonant sounds could be presented quickly or slowly, but they're okay with that task. Now up until this point, they've had the full consonant vowel consonant sounds, so dad, bad, gad. And these are recorded sounds. They're not synthetic stimuli. So there are slight differences in the duration of the stimulus, slight differences in the pitch of the stimulus, and rats are super smart, and we know that they can key in on some of these other cues. So to make sure that they were actually doing the consonant task and not relying on other cues, we removed all of the other cues. So for the final task, we presented only the first 40 milliseconds of the stimulus. So it's the consonant sound and the beginning of the vowel transition. But the stimulus length is now fixed at 40 milliseconds. And they fell apart again. So it seems like up until this point, the animals must have been using some other cue in the stimulus, like pitch or duration, to help them through these tasks. But the speech and noise proved too much of a distraction for those extra techniques to come in handy for them. And we know this is a pattern that's very consistent in individuals with dyslexia. So for the sake of time, I won't present all the rest of the data, but I will give you a little bit of a teaser that we tested another group of animals on the truncated speech task first. We gave them 30 days of training on just the phoneme discrimination, and after that, they did not have any differences in the speech and noise task. So it seems like the order of training matters, and focus on phonemes is very important on generalizing to other more difficult tasks. So following this 4 months of training, we then recorded from auditory cortex again. And in the black, you see the untrained animals. So again, this is the latency variance in the response. And you'll remember from before that the untrained Kia knockdown had a significantly higher variance. However, after training, we've normalized the timing of the responses so that they're no longer different from controls. Not only is the variance returned to control levels, but consonant encoding has also improved. So prior to training, we saw that the classifier performed worse using data from the Kia knockdown brain. Following training, there's no longer any difference between the Kia knockdown brain's ability to encode consonant sounds compared to controls. Then to tie a bow on the entire thing, we wanted to bring it back to the human world. And so this is the LFP response I showed you earlier in the A1 of the knockdown animal, the dark line, and the control in the light line. And following training, there again was no longer any difference in the LFP response of the knockdown compared to the control. So this is as close as we can get, again, to the type of techniques we use in humans, EEG, MEG. But these data suggest that we should be able to use these techniques to evaluate training efficacy in humans. So to summarize from this first experiment, suppression of Kia 0319 does increase variability in the neural response to speech sounds, and the amount of suppression predicts the degree of behavioral impairment. And this gene could be creating inconsistent neural representation of phonemes, which might be feeding the letter-to-sound correspondence problems that we see in dyslexia. But of course, we want to know, is this seen in humans as well? Does our research in rats translate into humans with this disorder? And that brings us to experiment 2 for today, to determine whether neural inconsistency is seen in the cortex of children with dyslexia. Now, recently, Nina Krause's group at Northwestern showed that children who are poor readers do have inconsistency in the auditory brainstem response to sound. So if you present synthetic stimuli over and over again, and you record the auditory brainstem, if you look at the consistency in the response across repeats, you see that children who are poor readers have significantly less consistency in the response compared to children who are good readers. So is this a feature that's also seen in cortex, or is it restricted to the auditory brainstem? Does the brain have some way of resolving this issue before it gets to cortex, or is it universal? So we're currently enrolling 40 kids between the ages of 7 and 16. Our goal is 20 with dyslexia, 20 typically developing. I'm happy to say that as of today, we've just hit the 20 with dyslexia mark. We do a behavioral battery with these kids, which I'll discuss on the next slide. We're doing saliva samples for genetic testing, and we're also using magnetoencephalography to record neural responses. So why MEG? MEG is a very non-invasive neural technique that measures the magnetic fields in the brain. So we all know that the Earth has a magnetic field. Our brains also have a magnetic field. Granted, it's about 1 billion times weaker, but it is there. And so this technique records and measures the differences in the magnetic field in the brain in response to sound. And so while many studies of dyslexia use EEG, which is the electrodes on the scalp that you've probably seen pictures of, you do get millisecond precision in both techniques, but what MEG gives us that EEG does not is spatial precision. So with MEG, we can get to within about a centimeter, of the source localization. So we'll be able to tell where the information is coming from, where the signal is coming from. Is it auditory cortex? Is it visual cortex? Is it somewhere else? We'll be able to tell with this technique. So in the MEG, they hear a variety of auditory stimuli, 4 different speech sounds, bad, dad, gad, and tad, and these are the exact same stimuli that our animals heard, so we can get as close to a direct comparison as we can. A variety of tones, so we have a non-speech auditory control. They also see a variety of letters on the screen and a variety of objects that don't really look like much of anything for, again, a non-language visual control. So our group characteristics, today I'm presenting data from 9 typically developing individuals and 7 kids with dyslexia. All of our kids are matched on nonverbal intelligence using the RIAS, and their standard scores all must be above 85. We're also controlling for oral language using the SELF-4. Since specific language impairment does co-occur with dyslexia, we wanted to make sure that our sample was pure reading disability. Our dyslexia group was diagnosed using the Tower 3 and the Wormit 3, which are 2 measures of sight word reading and phonemic decoding, and their standard scores had to be less than 90 to qualify. And so if we look at our group characteristics so far, they are, again, matched on the 3 areas that we expect, and then significantly different in their reading ability. So before we look for the variability in the auditory response, again, we want to validate that we're seeing the basic auditory processing differences that are characteristic of dyslexia. So using the auditory cortex response, we look at the response to all of our 4 speech sounds. We have our typically developing individuals in blue and our dyslexic individuals in red. And so the first marker is the M100. So this is right here, and this is a typically replicated marker of auditory response using MEG. And then this next box here, this is the M150 through the M300, and this is a well-replicated response of opposite polarity from the M100 that you typically see in speech sound processing. So at the M100, we see something that's a little bit surprising. We see our dyslexic individuals have a higher amplitude at the initial response, which again, given pilot data, the effect may go away, but it's interesting nonetheless. And then in our second window here, we see what we expect to see. We see that the amplitude in the dyslexic individuals is less than the amplitude of our typically developing individuals in response to sound. So now we want to go after our measure of neural consistency. So we have about 50 repeats of each of our stimuli. And first, we identify channels that are originating in the auditory cortex versus the visual cortex. We pick the 10 channels with the strongest response to a non-speech stimulus using the M100 response. So we look for responses to tones, and we grab the 10 channels that respond most strongly to tones. And the 10 channels, okay, so this is auditory, sorry, I won't talk about visual today. These are unique to each subject due to the location in the scanner. So in the scanner, there's pretty much this big suspended helmet, and a child's head could be anywhere within that scanner. So the channels that are grabbing auditory information from one child might be different than channels grabbing information from auditory cortex from another child. So these are unique to each subject. We then use the Pearson's correlation coefficient to compare similarity between every possible pair of responses. So, for example, we get an R value by comparing trial 1 and trial 2. Then we compare trial 2, trial 3. We compare trial 1 and trial 3, et cetera, et cetera, et cetera. And then we take the average of all of these R responses to get a single metric of neural consistency to each of our different stimulus types. So we can see that if we average across all stimulus types, there's the beginnings of a trend here. Of course, it's not significant with our numbers being as low as they are. But already we are seeing significant differences in stimulus type. So if you look at the consonant, vowel, consonant sounds and the auditory tones, there is already a significant difference in the consistency of the response in typical individuals compared to individuals with dyslexia. I don't have any genetic data to share with you today, unfortunately, but we will be presenting these data at SFN this year in the Wednesday morning block, so hopefully we'll see some of you there. Okay, so our preliminary conclusions, MEG responses in children with dyslexia confirm our previous reports of reduced amplitude in response to sound, and our current data suggests that neural inconsistency in response to sound may also exist in cortex of children with dyslexia. So I just want to close briefly by coming back to this idea of the symbiotic use of these 2 models. And we know that mice and rats are already being used to study genetics, the efficacy of pharmaceutical drugs, all of these things, but its use in language research is still not widely used. For example, FOXP2 is a gene that's very well known or was thought to originally be the language gene when it was identified in the KE family. But work in animal models, specifically in rodents, showed that this gene is not actually a processing gene. It's more likely involved in the motor pathway and in regulating other genes. Follow-up tests of that hypothesis in humans has shown that this gene and others in the same pathway are likely more involved in speech production than in speech perception. So I'd like to urge you to keep an open mind when we talk about using animal models to study language, not only on the genetic level, but also in the brain responses and also in behavior. And so thank you very much. I'm happy to take any questions.